Thank you for introduction. Because we are out of time, I will go quickly. Um, it's great honor for me to introduce our company, Celloid, and myself, and our technology. And if you are interested in organoid technology, you will like our production and products and technologies. So, yeah. So because Celloid is very small startup company, so let me introduce Celloid first. And we are a 3D circular solution provider based on South Korea. And actually, we are the majority of our team members are mechanical engineers rather than biologists, who has expertise in material processing and 3D fabrication and even robotics. What we are focusing on is organoid, which is the artificial miniature organ. Miniature organ, and what we are really doing is to build some tools, effective tools or platform that enables reproducible and controllable organoid culture, as shown in the below right side. So, so before we go into our technology, we need to talk about limitation of conventional organoid culture technology to understand our technologies. There are tons of 3D cell culture methods in academia, but these two, a hydrogen culture-based culture and a hydrodome-based culture and impermeable microwell is effective two major tools for 3D cell cultures. In the case of hydrogen dome-based method, they are really good for providing relevant environment for cells or organoids because they mimic the real human tissues, but it is really difficult to control the size or functionality of organoid within the hydrogel because it only it relies on the diffusion of material inside of the hydrogel layer. On the other hand, the microwell arrays are tools that has a very precise geometry so that it can it can enhance the uniformity and controllability of the organoids, but since they are made of plastic impermeable walls, so when the organoids grows in it, at the very bottom side of the organoid cells suffer from the lack of exchange of materials such as nutrient, growth factor, or even waste. So the status of organoids gets degraded as time goes by. And another practical limitation of conventional organic culture is that is from the long-term culture of organoid protocol. For example, the kidney, liver, pancreatic organoids takes a near a month, and for long period, for some organoids like brain, it takes even years. For such a long period, if we do exchange the media with our hand, the organoid faces the risk of contamination and organ loss, and this is a really important problem in a practical culture of organoids. So what we offer is two complementary solutions. The first one is the consumable, which named as a Nestwell, and this is the non-fiber membrane-based permeable microwell array. And yeah, because since it has microwell array structure, so it can provide you uniform organoids. And also it is made of permeable nanofibrous membrane, so it can enhance the diffusion of material near the organoids so that you can maintain the maturity of organoids. And the next solution is the organoid solution, which is the perfusion-based organoid culture automation system. This system utilizes the perfusion mechanism instead of the pipetting, so you don't need to open the culture chamber during the long period of the organoid culture protocol. So it effectively reduces the contamination risk or organoid loss. So we offer these two complementary solutions for your organoid culture. So I will go deeper more into the Nest2 platform, and this is the made of non-fibrous membrane as you left side, and this has 3D geometry shape for uniform size and uniform functionality of your organoid, and also it is since it is made of non-fibrous membrane, which is highly permeable, so you can maintain your organoid's maturity through this platform. As a proof of concept, we have conducted some kidney organoid culture on our platform, and as you see at the middle, you can see that the fluorescent image of kidney organoid, and the very left side there is an example of organoids grown within the hydrogel layer. You can see that we don't have any control on the size of functionality differentiation of the kidney organoid. But on the other, at the right side of the images, we can see very uniform size and uniform the organoids with uniform size and uniform geometries, and also we can control the size of organoids by changing the dimension of the dimensions of microwell. And at the very bottom, we have performed some full RNA sequencing analysis on our kidney organoids, and we found that the organoids from our platform have shown much similar cell composition compared to the human or mice kidney organ, compared to the organoids grown within the impermeable microwave such as agar-well. And also we could see that the organoid automatically generates the vascular, and we believe that it indicates that our platform provides the organoid multi-relevant environment for mature differentiation. So I will skip this page for the time. And the next, I will move on to the organoid system, which designed for automatic organoid culture. And our system consists of 3 independent modules. The first one is supply module, and this is a refrigerator for long-term media storage. And two small modules are perfusion module and vision module, and these are installed within the CO2 incubator chamber, so that you can grow the organoid within CO2 incubators. And the supply module has pump in it so that it can provide the media through the long and thin connecting tube between the tube connecting supply and perfusion module. And also this system utilizes the paperless, the perfusion-based media exchange mechanism, so it can reduce the contamination risk as shown at the bottom of the slide. Another important key feature of organized system is that it is not the multi-use bioreactor. It is actually a single-use bioreactor. So we provide the disposable cartridge for the system, which includes the 1 waste bottle and 5 media bottles, so that you can adapt it to your complex organoid differentiation protocol. And of course, this cartridge is provided sterilized and sealed. The cartridge composed of 2 separated parts. The one is for cell culture chamber and one is for media storage, and each part is installed to the system so that it can start the automatic organoid culture. At the 6 independent culture chamber of cartridge, you can set up the Nest2-based organoid production setup, especially for who want to produce or generate uniform size organoid. And for the researchers who want to directly adopt this system directly to their protocol, this system can be applied to the hydrogen-based culture as shown at the right side. So far, we Celloid have dedicated to enable some reproducible and controllable organic culture, and we have developed many products lined up for research use or high throughput drug screening or even large scale organoid culture. So if you have any interest on our product or technology, please contact me on this conference symposium. Thank you.